Welcome to the deep dive. This is where we take a whole stack of sources, sometimes complex ones, and really distill them down into the key insights for you. Today, we're diving into something that affects, well, millions of people. Dental implants.
Hmm.
You probably know someone who's had one, or maybe you have yourself. And you know their success, it really all comes down to the bone, right? The health of the bone.
Absolutely fundamental.
So what if that very first step, preparing the bone itself, is maybe about to get a major upgrade? We're looking at a really interesting new approach today. It's called the magnetic mallet technique for preparing the site for a dental implant.
Yeah, it's quite innovative.
And this deep dive, it's based on this fascinating pilot study. It was done in animals. The title is Biomolecular, Histological, Clinical, and Radiological Analyses of Dental Implant Bone Sites Prepared
Yeah.
using Magnetic Mallet Technology. Quite a mouthful.
Yeah, typical scientific title.
Right. So our mission here is basically to pull out the most important bits from this study, try to understand the biology, what's happening under the surface, and figure out what this tech could actually mean for, how well implants work in the future.
It really is at the kind of the cutting edge of how we're thinking about bone repair, bone regeneration. Lots to unpack.
I agree. So let's start there. Let's unpack that foundational point. Why is preparing the bone so critical for dental implants? Why does it matter so much for like the healing and making sure it stays stable long term?
It's a great place to start. It's like um, building a house, right? You need a rock solid foundation, otherwise nothing else matters.
Right.
For dental implants, the implant itself needs to fuse directly with the bone. That's the process we call osseointegration.
Osseointegration, okay.
Exactly.
Yeah.
And the current surgical techniques, well, they're always being refined. We're constantly looking for ways to be, less invasive, speed up healing, reduce trauma for the patient, offer the surgeon more control, and improve stability, both right away. That's primary stability, and down the line, secondary stability.
Mm.
And understanding the biology behind it all, things like inflammatory mediators or these bone morphogenic proteins, BMPs,
Yeah.
that's super important because they're kind of the managers of that whole healing process.
Okay, so BMPs are key players. So if traditional drilling, the way it's usually done, has maybe some limitations or challenges, what new ideas are out there for this crucial first step? Is there something really different emerging?
Well, that's exactly what this study is getting at, is looking at this pretty novel instrument called the magnetodynamic mallet.
Magnetodynamic mallet.
Okay,
Yeah.
and what makes this specific research stand out is that before this, most of what we know about this mallet was just from like doctors observing how it worked in practice, clinical
Right.
studies.
Oh,
Nobody had really dug into the histology,
Yeah.
Looked at the actual tissue under microscope or the biomolecular side, the cells and molecules involved. Not with this mallet.
So this study gives us that deeper look.
Precisely. It's a new level of insight.
That sounds fascinating. And the study, it had a very specific question it was trying to answer, right? A
Okay.
core question.
It did. They used the PICO framework, patient, intervention, comparison, outcome. Very standard in research.
Right.
And their question was basically, does using this magnetodynamic mallet technique to prepare the implant site, compared to just using the conventional drilling technique, actually improve how well the implant integrates with the bone osseointegration?
Okay, clear question. So how did they actually test that? How did they set up the comparison in practice?
So they used 3 male adult minipigs.
Minipigs. Why minipigs?
Yeah, they're actually a pretty common model in dental implant research.
Mm.
Their bone physiology is surprisingly similar to humans in many ways.
Yeah.
And interestingly, they didn't do the implants in the mouth. They used the shin bones.
Oh, okay. Why the leg bone?
It just makes it easier to standardize everything.
Yeah.
Control the conditions, the forces, without the complexities of the oral environment, like chewing.
Ah, gotcha. Less variability.
Exactly. So in each minipig's tibia, they prepared 2 sites using the magnetic mallet and 2 using the standard drilling method. That way they had a direct comparison within the same animal.
Okay, that makes sense. Smart setup. And the specific tools, what kind of mallet bits or drills did they actually use?
They had a set of specific mallet inserts, different sizes,
Yeah.
PF10, 160, F200, F230F, and they used specific power settings like power 2 to 4 on the device.
Okay.
And for the drilling, it was a standard sequence, like a lancelet drill, then maybe a 2.2 millimeter, then a 3 millimeter drill run at about 1,300 RPM. Pretty typical stuff for implants.
Right, so controlled conditions for both.
Yep. The idea was to make it a fair comparison.
And how did they measure the outcome? What were they looking at and when?
Okay, so key measurements. First, primary stability. They measured this using ISQ implant stability quotient.
I ask you outright.
They measured that right after placing the implant, that's TO times 0. Then they measured again 14 days later, T14.
14 days. Why 14 days?
Well, that 2-week mark is actually a really important time point. You can already see significant changes happening in terms of stability and the very beginnings of that bone integration process.
Okay. Makes sense. What else did they measure?
They also took x-rays and CT scans, computed tomography scans.
Ah, the 3D view.
Exactly. And then, crucially, at that 14-day mark, T14, they collected bone samples from around the implant sites.
Right into the lab.
Yep. For those detailed histological tissue level and biomolecular cell and molecule level analyses, that's where the really deep story is.
Okay, so let's get into that story then. The bone story. What did they actually find when they looked super closely at the bone prepared with this magnetic mallet?
This is where it gets really interesting, I think. The histology, looking at the tissue slices, it showed a significant increase in the area of newly formed bone around the mallet-prepared sites.
Wow, okay. More bone.
And not just more bone area, but also a significant increase in the number of osteoblasts.
Those are the bone-building cells, right?
Exactly, the builders. And here's another key thing. Those mallet sites also showed less fibrous tissue, sort of like scar tissue. Now, that fibrous tissue finding wasn't quite statistically significant. It was more of a trend, but it strongly suggests a better healing environment overall. Less scar, more functional bone.
Okay, so it's not just more bone. It sounds like it might be better quality bone, or at least heading in that direction.
That seems to be the implication, yes.
So what about the biomolecular stuff, the signals? Did they see specific bone-building signals being turned up?
They did. The biomolecular results really backed up the histology. They found a statistically significant increase in BMP4 in the mallet sites.
BMP4, that was one of those bone morphogenic proteins you mentioned.
Yep, that's the one. BMP4 is like a really potent signal. Think of it as a recruitment officer.
Okay.
It calls in these special cells, mesenchymal stem cells, and basically tells them, right, your job now is to turn into osteoblasts and start building bone.
Wow, so it's actively recruiting the builders.
Pretty much. And they also saw upward trends, though not statistically significant at 14 days, for other helpful factors like BMP7, TGF2, RUNX2, OCN, all things that generally encourage bone formation.
That's amazing. It's like the mallet isn't just making space, it's sending out memos telling the bone exactly how to heal better.
That's a good way to put it, yeah. It seems to be triggering a very specific biological response.
But I noticed something in the study that seemed, well, a bit weird, maybe counterintuitive. The levels of ALP and CoL1A1 were actually lower in the mallet sites. How does that fit in if there's more bone being formed?
That's a really sharp observation. It does seem like a bit of a paradox at first glance, doesn't it?
Yeah.
But the study proposes a pretty logical explanation. The hypothesis is that the osteogenesis, the bone-building process, might actually be happening faster or starting earlier in the mallet sites.
Ah, okay, so maybe they peaked before the 14-day measurement?
Exactly. So factors like ALP, alkaline phosphatase, and CoL1A1, that's collagen-type IL-alpha-1, they might have already hit their highest levels and started coming down by the time the samples were taken at T14.
So the measurement timing is key. It might mean the healing is actually further along.
It could, yeah. And the lower CoL1A1, the collagen marker, that ties in really nicely with seeing less fibrous tissue.
Because fibrous tissue is mostly collagen.
Right. So less CoL1A1 could simply mean less of the less desirable fibrous tissues being laid down. It all kind of fits together.
That makes sense. Okay, that clears up the paradox. What about inflammation? You mentioned inflammatory mediators earlier. Was there a difference there between mallet and drill?
Yes, there was. The mallet sites actually showed higher levels of some pro-inflammatory cytokines, things like IL-1, IL-6, IL-8, TNF.
Oh, so more inflammation. Isn't that usually bad?
You'd think so, but here's the crucial part. It looked like this inflammation was under control.
Under control. How?
Because at the same time, they also saw increased expression of anti-inflammatory mediators, specifically IL-10 and PGF2.
Ah, so the body was also sending signals to calm things down.
Exactly. It's just a balanced response. Like, the initial inflammation might be necessary to kickstart the healing, clear debris, but the body is also actively regulating it, preventing it from getting out of hand and causing damage. It's productive inflammation, if you like.
Okay, a controlled demolition to clear the way for new building. I like that analogy.
Yeah, something like that.
And the study also mentioned something called the want pathway. Can you briefly explain what that is and how it fits in?
Right, the WNT pathway. Think of it as one of the body's master control systems. It's involved in loads of things: cell growth, development, and really importantly for us, bone formation.
Okay, a master controller.
Yeah, it basically helps tell cells what to do, grow, divide, become specialized. And in the Mallet sites, they saw some really interesting changes in which parts of this WNT pathway were active.
Like what?
Well, for example, they saw lower levels of something called Win3A, but increased levels of 1A5A, 1A210B, and 116.
Okay. And what does that translate to biologically?
So simplifying a bit, 1A can actually put the brakes on 1,3A sometimes. 116 helps block the cells that break down bone, the osteoclasts.
Okay, stopping bone resorption.
Right. And 1N10B directly triggers cells to become bone formers, the osteoblasts.
Mm.
So the overall pattern seems to shift the balance towards more bone formation and less breakdown.
Wow. So it's influencing these really fundamental control pathways.
It appears so. And interestingly, that increased 1,5A can also be linked to some of those increased inflammatory markers we saw earlier. It's all interconnected.
It really is like a biological symphony being orchestrated by the mallet. Incredible detail. Okay, let's shift gears slightly to the more sort of clinical side. What about implant stability, the ISQ numbers?
Right, the practical outcome. So here's the finding. For both techniques, mallet and drill, they saw a significant increase in stability, the ISQ score, between day 0 and day 14.
Okay, so both methods led to more stability over 2 weeks. That's good.
Yes, but, and this is really key, there was no significant difference in the ISQ values between the mallet and the drill techniques at either time point, T0 or T14.
Wait, no difference in stability. But you just described all these biological advantages for the mallet: more bone, better signals. How does that work?
Exactly. It's the crucial point to unpack. The raw stability numbers, the ISQ, were similar, but how that stability was achieved seems to be different.
Okay.
The study suggests that the stability seen in the mallet sites is probably due to something called osseocondensation.
Osseocondensation, compacting the bone.
Precisely. The mallet technique seems to compress and condense the softer, spongy, cancellous bone sideways, packing it in around the implant site, making it denser right from the start.
Ah.
And they actually noted that the insertion torque, how much force it took to screw the implant in, was higher in the mallet sites. That supports the idea that the bone was denser, more compacted.
Right, more resistance.
Whereas the drill, by its nature, removes bone to create the space.
Yeah.
It's designed differently, though it still achieves good mechanical stability.
So similar stability scores, but achieved differently. One by compacting existing bone, the other by creating a precise hole in it.
That seems to be the interpretation, yes. The quality or nature of the bone providing that stability is different.
Did they actually see this condensation, like on the scans?
They did. The CT scans were apparently quite clear on this.
Oh, yeah.
They showed, and I quote roughly, a moderate trabecular densification organized at the cortico-cancellous junction, basically. Denser spongy bone right where it meets the hard outer layer next to all the implants put in with a mallet.
Wow.
And this lateral bone condensation, as they called it, was mostly missing in the sites prepared with the drill.
So clear visual proof of the mallet's unique action.
Exactly. Really compelling visual evidence.
Now, were there any downsides? Any sort of practical challenges or limitations mentioned with using the mallet?
Yeah, they did note a couple of practical points. The mallet device was apparently less performing, as they put it, in really dense, thick cortical bone, that hard outer shell.
Okay, so maybe not ideal for super hard bone.
In those cases, they suggested maybe needing a little bit of pre-drilling just to break through that toughest outer layer first.
Makes sense.
They also gave some technique tips, like starting the mallet on a lower power setting and then increasing it gradually. This helps avoid, potentially damaging the delicate spongy bone structure.
Right, ease into it.
And also, while the mallet itself doesn't need constant water irrigation like a drill does, they recommended maybe rinsing intermittently with some sterile saline.
Why is that?
Just to keep the bone tissue a bit more elastic, potentially reducing the chance of tiny microfractures. Though, importantly, they stressed that they didn't actually observe any fractures in this particular study.
Okay, so some technique considerations, but nothing majorly problematic in this setup.
Seems that way. It's more about understanding how to use it best in different situations. But overall, the study really underlines its effectiveness for preparing the site and, crucially, for how it influences the bone biology.
So not quite a magic wand for every single bone type, but used correctly, it's highly effective and promotes this really healthy bone response.
Exactly. That's the main message.
Okay.
Despite the practical points, this study clearly shows it works well, especially in promoting that healthy bone formation and condensation.
Well, this has been a truly fascinating deep dive. We've really unpacked how this magnetic mallet, even though it might show similar stability numbers initially compared to drilling, it seems to have this profoundly different impact on how the bone heals right down at the biological and tissue level, promoting more bone, better formed bone, through this osteocondensation and by
Yeah.
tweaking those key molecular pathways.
Yeah, it really does seem to represent a shift in thinking.
It feels like it could be opening up a new chapter in how we approach preparing for dental implants.
And, if we just zoom out for a second, connect this to the bigger picture,
Yeah.
think about what this implies, this deeper understanding of how bone responds to different kinds of mechanical force, different surgical approaches, and the potential ability to steer specific biological pathways towards better healing. That could have implications far beyond just dental implants, couldn't it? Think about wider bone repair, regenerative medicine, maybe even how we approach recovery from injuries in general.
Wow.
What other areas might benefit if we could achieve this kind of precise control over the bone's own healing environment? It's something to think about.
That is a really provocative thought to end on. Where else could targeted physical stimulation enhance biological healing? Lots to ponder there. Well, thank you so much for joining us on this deep dive today. It's been incredibly insightful.
My pleasure. Fascinating topic.
And thank you, our listener, for joining us. Until next time, keep exploring the cutting edge.